Good morning, dear students. This is your teacher Neelam Elawat, and we are doing the chapter 10 Cell Cycle and Cell Division. I hope you've understood the topic that we finished yesterday. Yes, that is. Which topic we did yesterday? Exactly. We talked about the cell cycle, the different phases of the cell cycle, right? Now, in today's video, dear students, we'll be talking about the mitosis process. Okay, it is basically a type of cell division, right? It's basically a type of cell division. Okay, and this only occurs in case of somatic cells. So now in this particular lecture today, we'll be talking about the cell division process, which is mitosis, and it only occurs in the somatic cells, somatic diploid cells. Diploid, having double number of chromosomes, that have double number of chromosomes. So diploid means having double number of chromosomes. So mitosis basically occurs in the somatic diploid cells, right? Okay. But in case of plants, dear students, the mitotic divisions, it occurs in both. In diploids, in case of animals, it occurs in the diploid, somatic diploid cells, while in case of plants, this occurs in both haploid as well as diploid. Okay, this occurs in both haploid as well as diploid cells. In case of animals, it only occurs in the somatic and the diploid cells. So as I told you, students, it is a cell division that occurs in the somatic cells in case of animals, and it occurs in somatic diploid cells in case of animals, while in case of plants it occurs in both diploid as well as the haploid cells. Now when a cell divides, when a cell divides, suppose we have this cell and then this cell divides to form the two cells. In these two cells, the number of chromosomes will remain same. Means suppose you had 23 chromosomes here. Here also each cell will have 23 chromosomes. So in this type of cell division, the number of chromosome in the parent as well as the number of chromosomes in the cells, the daughter cells formed, remain same. So we call this division as equational division. Okay, why do we call it as equational division? Because the number of chromosomes that are present in the parent as well as in the daughter cells is same. That is why we call it as an equational division. Okay, that is why we call it as equational division. Right? And as I told you, it occurs only in the diploid and the somatic cells in case of animals, while in case of plants, it occurs in both of them, haploid as well as diploid. Now, during this phase, there is reorganization of all cellular components. Okay, बेटा, इस phase में जो है आपकी सेल के अंदर जितने भी components मौजूद हैं उसका reorganization होता है. Okay, right? And if you look at the picture, just see we have a single cell, right? Then the single cell, it will have the chromosome number will be doubled. It will pass through the different phases. Then it will divide to form the two cells, and ultimately there is a formation of two cells. So this type of cell division results in the formation of two daughter cells, and each daughter cell has the same number of chromosome as they are present in the parent. So that is why it is called as equational division. Clear? Okay. Now, as I told you, dear students, that the division phase is comprised of two phases. So what are those two phases? Tell me, everyone, what are the two phases? Yes, very good. One is your karokinesis. Cell division phase is comprised of two phases, karokinesis, and second is cytokinesis. So similarly, mitosis also comprised of two phases, karokinesis and cytokinesis. Now here karokinesis is comprised of four, it can be further divided into four stages. One is prophase, second is metaphase, third is anaphase, and the fourth is telophase. Okay, so what are the different phases? 4 phases: prophase, metaphase, anaphase, telophase. Now just look into the picture also. These are the prophase. This is early prophase. Okay, and this is late prophase. So prophase, then you have second phase is metaphase, third is anaphase, and fourth is telophase. Okay, everyone? Right. Now coming to the first phase. Okay, that is called as your prophase. So prophase is the first phase, right? And it comes after the G2 phase, right? If you remember, first we have G1 phase, then we have S phase, then we have G2 phase, okay? And then the cell will enter the M phase, okay? So this is the first phase that comes after the G2 phase. And it is the longest phase, okay? It is the longest phase. Now, as I told you, prophase can be divided into two, early. It can be divided into early prophase and then late prophase. Okay, everyone? It can be divided into two phases, early prophase and you can say late prophase. In early prophase, dear students, the chromatin fibers, they will condense to form the chromosomes. Okay, first of all, let's compare what is happening. Initially, like if you look at a cell, let's have a different color so that you can understand properly. Let's have this. Initially, the cells will be, the cell contains the chromatin fibers, entangled chromatin fibers. देखिए, अगर हम पहली बहुत ही अर्ली प्रोफेज में सेल को देखते हैं तो सेल के अंदर जो है थ्रेड लाइक स्ट्रक्चर्स मौजूद होते हैं, लाइक एज यू कैन सी हियर, एंड दीज थ्रेड लाइक स्ट्रक्चर्स दे आर कॉल्ड एज क्रोमैटिन फाइबर्स. ओके, इन्हें हम क्या कहते हैं? क्रोमैटिन फाइबर्स. ओके एवरीवन, वी कॉल देम एज क्रोमैटिन फाइबर्स. तो सेल है, सेल के अंदर जो है मैगी लाइक स्ट्रक्चर प्रेजेंट होता है, एंड दैट मैगी लाइक थ्रेड लाइक स्ट्रक्चर्स आर कॉल्ड एज क्रोमैटिन फाइबर्स. Okay, now then these chromatin fibers, they will arrange themselves to form the chromosomes. Now basically a chromosome consists of like this structure. The chromatin fibers, they will rearrange themselves to form chromosomes. Okay, they will rearrange themselves to form the chromosomes. And one chromosome is composed of two threads. These two threads, they are called as chromatids. What do we call them as? Chromatids. So in the early prophase, the cell contains thread-like structures. Those thread-like structures are called as chromatin fibers. Then these chromatin fibers, they will condense themselves to form the chromosomes. And one chromosome consists of two chromatids. As you can see here, one chromosome is comprised of two chromatids, and these two chromatids, they are attached with each other. They are attached with each other with the help of centromere. So in the center we have centromere. Okay, what do we have in the center? Centromere. So one chromosome is made up of two chromatids which are attached with each other with the help of centromere. Okay? Clear? Okay. I hope I am clear, everyone. So in the early prophase, there are thread-like structures. Those thread-like structures are chromatin fibers. Now these chromatin fibers, they will condense to form the chromosomes, and one chromosome is comprised of two parts, that is two chromatids, which are joined with each other by a centromere. Okay? Clear? Hai? Okay. Now in the late prophase, what will happen? As you see in this structure, as you can see, this is one chromosome. This is another chromosome. So there are basically 4 chromosomes. Now in the late prophase, okay, in the late prophase, we know that each chromosome has 2 chromatids which are attached at the centromere. still the condensation of the chromosomes, it will continue. Okay, the condensation of the chromosomes, it will continue, right? Now basically in case of animal cells, what happen? The centrioles, they will move to opposite poles, right? You will find here the centrioles, they will start moving to opposite poles. One will move to the north pole, another will move to the south pole, and they will start radiating the astral rays. They will start radiating the astral rays, right? Or their tubule formation will start, or microtubule formation will start. So the you can say the two asters along with the spindle fibers, they form the mitotic apparatus. So what is mitotic apparatus? What is mitotic apparatus, everyone? Mitotic apparatus is, what is it? Asters plus the spindle fibers. Okay, asters plus the spindle fibers. Okay, so mitotic apparatus. So centrioles, they will move to opposite poles and they will start radiating the astral rays, and these astral rays along with their centriole pair is called an aster. And the two asters, they will move to the opposite poles and they will start their spindle formation, right? Is it clear? Centrioles, they will move to opposite poles. One centriole will go to one pole. Now these centrioles, they will start radiating the astral rays. Now these astral rays along with their centriole pair is called an aster, and the asters, they will move to opposite poles and they will start the spindle formation. Or we can say we have a mitotic apparatus which comprises of two asters and the spindle fibers, right? Now along with this we will observe that the nuclear envelope will disappear. Okay, the envelope around the nucleus, that will start disappearing. Golgi apparatus will disappear. Endoplasmic reticulum will disappear. Nucleolus will disappear. So all these structures, they will start disappearing, right? And spindle fibers, they will start originating from the microtubular proteins. Okay, they will start originating from the microtubular proteins. Now after the prophase, the second phase will start, which is called as metaphase. What do we call it as? Metaphase. Okay, in metaphase, if we see, look at the cell, just look at the cell. Can you see the cell? Can you see? Yes. So in metaphase, the nuclear envelope has completely disappeared. There is no nuclear membrane around the nucleus, right? So nuclear membrane has completely disappeared. Okay, and the chromosomes are highly condensed in this stage, right? The chromosomes, they are highly condensed in this stage, and if you look at them, they consist of two parts. They are composed of two chromatins. Like if you see, they are like this. Just look at this. They are highly condensed, isn't it? They are highly condensed in case of metaphase. And just look at this. These are the two asters. Okay, these are two astrals and rays they are radiating. So these are spindle fibers. जो ये बन रहे हैं ये spindle fibers हैं. और आप देखिए दो आपके centrioles arranged हैं. इनको हम कहते हैं astrals. Okay. So अब इस केस में chromosome condensation जो है इस at metaphase stage, the chromosomes, they are highly condensed. Okay, and they can be easily observed under the microscope. They can be easily under the microscope, and you can say they are comprised of two chromatids which are attached with each other, and slowly and gradually these chromosomes, they will appear to come to lie at the equator. धीरे धीरे सारे chromosomes जो है वो आकर equator पे lie करेंगे. They will start appearing at equator, right everyone? They will start appearing to come at equator, as you can see in this cell, metaphase cell. The all chromosomes, they will arrange themselves at the equator, right? And this this arrangement of chromosomes at the equator is referred to as metaphase plate. What do we call it? Metaphase plate, right everyone? We call them as metaphase plate. So what is metaphase plate? The arrangement, the plane of alignment of the chromosomes at the metaphase. Okay, what is metaphase plate? The plane of alignment of the chromosomes at the metaphase is referred to as metaphase plate, right? Now, since the chromosomes, they are arranged in the center, like as if you look at this, one chromosome, I'm just talking regarding this. So this is a chromosome, it is attached in the center of the, it is attached to the equator in the center of the cell. So these spindle fibers, they will attach here. Okay. From both sides, the spindle fibers, they will attach with the chromatids, okay, by their kinetochores in the centromere. So दोनों साइड से स्पिंडल फाइबर्स जो हैं वो आकर आपके सेंट्रोमियर से काइनेटोकोर से अटैच हो जाएंगे, सेंट्रोमियर से अटैच हो जाएंगे, राइट? क्लियर एवरीवन? The spindle fibers from the both sides, they will attach to the centromere part of the chromosome. In the centromere, there is a part which is called as kinetochore. Okay, centromere जो है structure, that is inside it there is a part which is called as kinetochore. तो आपके सारे spindle fibers जो है वो kinetochore से attach हो जाएंगे. And now at this phase, the chromosomes, they will divide and they will start moving to the opposite poles. इस फेज पर क्योंकि both sides on from both sides, from the north pole also, from the south pole also, they are attached with the spindle fibers. So now the chromosome will divide. Okay, chromosome containing two chromatids, it will divide. देखिए chromosome है, chromosome के पास ये दो parts हैं, यहाँ से attached है, यहाँ से आपका spindle fiber इसके साथ attach हो जाएगा और एक यहाँ से हो जाएगा. तो ये जो chromosome है ये center से divide हो जाएगा. Okay, because it is being pulled from both sides. ये chromosome को जो है दोनों साइड से pull किया जा रहा है. That is why this chromosome, it will divide from the centromere, right? Now, as I told you, see this, the chromosomes that has divided, and they have now one chromatid is moving to this pole and one chromatid is moving towards this pole, right? So in the metaphase, when the chromosomes they are present at the equator and they are being pulled on the both sides of the chromosome, so the chromosome will divide from the center, and now the chromatids, they will move to the opposite poles, and that will be the beginning of the anaphase. Okay, once the chromatid, they start moving towards the opposite poles. So this is the shortest phase of the mitosis, where you will observe that the chromatids, they are moving towards the opposite poles. Okay, these spindle fibers, they will contract and they will pull the chromatids to the opposite poles, as you can see here. So this is the early anaphase. Now in late anaphase, as you can observe here, the chromosomes, they are reaching up to the different poles. And as you can see, they have V-shaped chromosomes. The shape of the chromosome, just look at this, right? The chromatids, they have become V-shaped. Okay. So in this phase, although the chromosome has split and chromatids, they are moving to the opposite poles, but we will not call them as chromatids, we will call them as chromosomes. They are future chromosomes. Okay. They are future chromosomes because in S phase, the DNA had became doubled. आपको याद है ना, एस फेस में हमारा डीएनए जो है वो डबल हो गया था, क्रोमोसोम का नंबर सेम था, राइट? सो यहाँ पर आपके जो क्रोमोसोम्स हैं, इट हैज स्प्लिट एंड वन क्रोमैटिड, वन पार्ट ऑफ द क्रोमोसोम इज मूविंग टुवर्ड्स वन पोल, बट वी विल consider them as the future chromosomes. Okay, clear? So as the chromosomes, they reach to the poles, there will be starting of the next phase, which is called as a telophase. Now in this phase, all the chromosomes that are reaching up to the poles, they will uncoil and again they will appear in the form of chromatin fibers. ठीक है ना? अब आपके क्रोमोसोम्स जो हैं ऑपोजिट पोल्स पर पहुंच जाएंगे. ये क्रोमोसोम्स जो हैं बेटा, ये वापस से अनकॉयल हो जाएंगे, वापस से डीकंडेंस हो जाएंगे, एंड दे विल नाउ अगेन अपीयर इन द फॉर्म ऑफ क्रोमैटिन फाइबर्स. एंड सेकेंडली, a nuclear envelope will start appearing around these chromatin fibers. और एक nuclear envelope है जो इन chromosomes के around formation उसकी start हो जाएगी. न्यूक्लियोलस रीअपियर हो जाएगा, गोल्गी apparatus दोबारा से रीअपियर हो जाएगा, endoplasmic reticulum will also reappear, and the spindle fibers, they will disappear. Okay, there is a disappearance of the you can say the spindle fibers. So chromosomes, once they reach to the opposite poles, they will uncoil and they will appear in the form of chromatin fibers. Then nuclear envelope will, start forming. There is a formation, a reappearance of the nuclear envelope along with the different cell organelles, and the spindle fibers, they will disappear. So with this last phase, telophase, the first step of the M phase will finish, that is karokinesis. Now we will have the second step, which is called as cytokinesis. Okay. Now we will have a second phase which is called as cytokinesis, means the division of cytoplasm. What is cytokinesis? It is division of cytoplasm. Okay. Now this step is basically different in case of plant and animal cells. Now first we discuss about the animal cell. Okay. What happens in an animal cell? There is a cleavage, there is a formation of a cleavage furrow. Okay. From outside to the inside. यहाँ से एक furrow reappear होना शुरू हो जाएगा. As you can see, this is a furrow. Now this cleavage furrow, it will divide the single cell into two cells. Clear? So in cytokinesis, in case of animal cells, the formation of furrow will start from the outer side. यहाँ पे जो furrow की formation हो रही है बेटा जी, वो outer से inner side में हो रही है. It will move from outer to the inner side, and gradually the cytoplasm of the cell will divide into two equal parts. और दोनों cells के पास जो है equal आपकी cytoplasm divide हो जाएगी. So the basic difference that we have to note is that cleavage furrow appears in the plasma membrane, and you can see it will move from outer to the inner side. Now talking about the cytokinesis in case of plant cells. See, look at the picture, first picture. As you can see here, the cytokinesis is different in case of plant cells. Why? Because they contain cell wall. In animal cells, there is no cell wall, but in case of plant cells, we have cell wall. So what basically will happen, how the cytoplasm will divide in case of plant cells? First of all, the Golgi apparatus, it will release vesicles. Okay, what will happen? The Golgi apparatus will release the vesicles, and these vesicles, they will arranged in the center. Okay, समझ आ रहा है बेटा? जो गोल्गी apparatus है वो vesicles रिलीज करेगी, और vesicles जो है वो अपने आप को सेंटर में अरेंज कर देंगे. ठीक है? As you can see here, there is a formation. See, the vesicles, they are arranging themselves in the center, and this arrangement is occurring from inner to outer side. देखिए सबसे पहले जो vesicles arrange हो रही हैं वो बिल्कुल सेंटर में हो रही हैं, और सेंटर से फिर वो बाहर की तरफ आ रही हैं. Okay, so here the arrangement of vesicles is occurring from inner to outer side of the cell. Okay, so here they will form a plate. This growth is referred to as a cell plate is being formed. Now this cell plate will divide the two cell. This cell plate will divide the cell into two equal compartments, right? And this is occurring from inner to outer side, as you can see here, right? And once the cell is being divided, okay, the cell is dividing. So all the cell organelles like mitochondria, plastids, they are equally distributed between these cells. ठीक है, जब जैसे जैसे आपकी सेल प्लेट की फॉर्मेशन हो रही है बेटे, तो सेल के अंदर जितने भी सेल ऑर्गेनल्स मौजूद हैं उनको इक्वल डिवाइड किया जा रहा है दोनों साइड पर, राइट? ओके, clear? And this will result in the formation of a cell plate, and this cell plate will divide this single cell and will form two daughter cells. Now once this there is a division of the cell, then this cell plate, it will form the middle lamella. Okay, this will form the middle lamella, the middle connection between the two cells. Now in case, see, in most of the cells after the karyokinesis there is cytokinesis, but in some cells there is no cytokinesis after the karyokinesis. तो अगर cytokinesis यानी साइटोप्लाज्म डिवाइड नहीं करता है न्यूक्लियस डिवीजन के बाद, तो उस केस में क्या होता है, मल्टी न्यूक्लिएट कंडीशन प्रेजेंट होती है. सिंगल सेल means a single cell will contain many nuclei if its cytoplasm does not divide, and this is the case that we observed in case of liquid endosperm which is present in coconut. So coconut में बेटे आपको यही कंडीशन देखने को मिलती है, वहाँ पे आपका न्यूक्लियस डिवाइड हो जाता है लेकिन साइटोप्लाज्म डिवाइड नहीं होता है. So that is why you have many nucleus which are present in a single cell. So you have a multi-nucleated condition. and that you see in case of the liquid endosperm, which is observed in case of coconut, right? I hope everybody has understood, bete. Okay, so can you tell me what are the two steps that we have done? Right, chirokinesis and cytokinesis, right? Okay. Now these are the overall steps. Just look at this. First of all, we had the interphase, isn't it? Right? So first phase is the interphase. Then second is the M phase. M phase is your mitotic phase. In M phase, the first phase is called as prophase. Right? Then we have a metaphase. Then we have anaphase. Then we have telophase. And then we have cytokinesis. And cytokinesis will divide the one cell into two daughter cells. Right? Is that clear, everyone? Is that clear? Yes. Okay. So these are the overall steps of the mitosis. Okay. And in mitosis, as I told you, same number of chromosomes are present. So this is also called as equational division. Okay. What do we call it as? Equational division. Now, why do we are talking about the mitosis? Why the mitosis is important? Why do we think that a cell need to divide? क्या ज़रूरत है एक सेल को डिवाइड करने के लिए? तो माइटोसिस, व्हाई द माइटोसिस प्रोसेस इम्पॉर्टेंट? फर्स्ट ऑफ ऑल, एज आई टोल्ड यू, इट प्रोड्यूसेस डिप्लॉयड डॉटर सेल्स. जो नए डॉटर सेल्स बनते हैं बेटा जी, दे एग्जैक्टली हैव सेम नंबर ऑफ क्रोमोसोम एज देयर पेरेंट. सो इफ द पेरेंट इज डिप्लॉयड, okay, if the parent is diploid, the new daughter cells, they will also be diploid. So this process results in the production of diploid cells. Secondly, it helps in maintaining the same number of chromosomes in all the somatic cells. Third, suppose if your body is damaged, okay, then your body needs to be repaired. And for repairing the cells, we require new cells. Isn't it? अगर हमारी बॉडी कहीं से डैमेज हो जाती है तो हमारी बॉडी को वहाँ पर नए सेल्स की ज़रूरत पड़ती है. तो नए न्यू सेल्स की फॉर्मेशन कैसे होगी? माइटोसिस से. ओके, सो माइटोसिस बेसिकली हेल्प्स इन द रिपेयर एंड रिप्लेसमेंट ऑफ द सेल्स. ओके, लाइक इट हैपन्स इन योर ब्लड सेल्स, इट हैपन्स इन योर स्किन. सो योर स्किन इज बीइंग रिन्यूड. द नंबर ऑफ RBCs, they are being renewed, right? Because we know the life cycle of RBC is 120 days, right? So जो हमारी बॉडी में रिपेयर हो रहा है, रिप्लेसमेंट हो रहा है, that is all because of the process mitosis, right? Okay. Now if you look at if we talk about the plants, we know that in case of plants there are meristematic tissues are present. Okay, do you remember? Plants contain meristematic tissue, the dividing tissue. Yes, and this meristematic tissue is present at specific places. That is why the growth of the plant occurs at specific points. Do you remember? We have apical meristem, lateral meristem, intercalary meristem. So that is why, because plants have meristematic tissues, that is why they grow continuously. Okay, the growth of plant occurs continuously at specific points, right? And the most important, everyone, mitosis basically helps to restore the nucleus and the cytoplasmic ratio. Because first the nucleus divides, then the cytoplasm divides equally. So there is equal division of the nucleus as well as the cytoplasm. So generation after generation, the nucleic and the cytoplasmic ratio remains same. Okay, the ratio remains same, right? So with this we finish our topic, dear students. Now what, I hope you've understood and there is no queries regarding this. Even if you feel, just go through this topic, just revise this topic, right? That are the page numbers 164 to 137. Just read it thoroughly, underline the important points. There are certain words which are highlighted with black. Don't forget to, underline them and understand them. And if there is any query, anything that you feel there is some queries or doubt, you can discuss it with me personally. And most important, make notes of this topic. Okay, so you have to make notes of the topic. You have to read these page numbers. And if you find there is some problem, you can discuss, right? Okay, bye everyone.